So, chemical reactions to get us all this wonderful, fabulous ATP going. Now let's look at the actual pathway to which we get all this mythical ATP. To get all this mythical ATP, we are going to undergo the process of cellular respiration. It's a energy yielding pathway. It is a number of steps. There are 1, 2, 3, 4 kind of major parts to cellular respiration, and within those, the vast majority of them have several parts, several steps in this pathway to yield all of this ATP. You can make ATP without energy. Does anybody know what the name of that one is? That would be anaerobic. With an anaerobic pathway, it's done, and means without. So, I mean, you're never without oxygen, right? I guess if you are without oxygen, you're dead. So you can be in a state of anaerobic in terms of the literal definition of the word, but anaerobic, just not enough oxygen is present. When it comes to this, we use anaerobic metabolism, which is lactic acid, the Cori cycle. With anaerobic respiration, we do not involve the mitochondria because the mitochondria requires oxygen to get inside and do all the fun stuff that we do inside of a mitochondria. With anaerobic, it's just happening within the cytoplasm. Okay, so it's just going to be glycolysis only, the first step that we'll talk about here in a second. But we can function under an anaerobic state, not for an extended period of time. Okay, not for an extended period of time. But if you've ever worked out high intensity interval training, HIIT training, or Tabata type training, and you go balls out for 20 seconds, a minute long, and take a 10-second, 20-second break, and then go back at it again, you've been in this anaerobic state. Okay? Your body utilizes the lactic acid that it produces, sends it from the cell into the blood to the kidney, to the liver, to convert it back into glucose, back into the blood, back into the muscle, and you can make energy. Obviously, this is not a big yielder of energy. It's like only 2% efficient. Okay, you only make like 2 ATP. But it can sustain you, and you can keep going. Not for perpetuity, but you can at least keep running from the bobcat. Okay. So when we talk about this, we are talking about the use of oxygen. Okay, glucose oxidation. Again, primarily, we're just talking about glucose. We're not talking about beta oxidation, the utilization of fatty acids. We're not talking about the utilization of protein and being able to yield this energy. We're just going to be talking about glucose. Okay. Again, that's glucose's primary function, is to provide the body with energy. All right. You start with this big old molecule called glucose, bunch of bonds, lots of potential energy, mwah, yummy. And we break it down into their basic component parts, ultimately down to carbon dioxide and water. And those things, from an energy standpoint, nothing to them. Water is removed and transported out of the body. Carbon dioxide is a waste product as well. It's transported out of the body. Exhale, carbon dioxide. Sweat, urinate, water removed. All right. There is the chemical reaction down there for it. C6H12O, C6H12O6. This is glucose. That is oxygen. There is my carbon dioxide. And then there's my water. Question? Heavy breather?
I almost asked that question and you changed it.
Yeah, beat you to it. C6H12O6.
Yes, glucose.
Yes, sir. All right. Okay. Oh my gosh. Sorry, my system gave me a weird prompt right there after I was just sighing really loud. Yeah, let's just, yeah. This is what we're talking about right here. Okay. Utilization of oxygen to make ATP. That's all that word here. That's all this word means. Okay. That's all this means. Utilizing oxygen to add a phosphate. Okay. To add a phosphate. That's all that stuff means. Okay. So if you have some kind of semblance or mastery of the English language, you've got science. Okay. Break a word down. What does it mean? That is what its function is more than likely going to be. A buttload of enzymes, as you might imagine. I'm not going to ask you those because that is a biochem class and this is not biochem. Okay. So we're going to talk about cellular respiration. I need you to know where these steps occur. So this would be where you write down. Where do these occur? Glycolysis, transition reaction, Krebs cycle, electron transport chain. And when it comes to where these occur, there's only two parts, cytosol or mitochondria. And obviously we're going to get more specific when we get into the mitochondria. But these are the two general areas that we have. If we don't have oxygen, the doors are closed to the mitochondria. We have to have oxygen available to get inside the goodie bag of the mitochondria. No oxygen, no mitochondria, no big yield of energy. And then we're in an anaerobic state. So we have four major steps in the formation of ATP. Okay. We have the glycolysis, transition reaction, which this book calls the intermediate stage. You can call it, especially if I ask it as a short answer, because this makes a good short answer question, does it not? Of course it does. Multiple steps, lots of stuff. You can easily screw it up. Salivate. Okay. Glycolysis, intermediate stage or transition reaction. Okay, I learned it as transition reaction. Most books have that. This one has it as an intermediate stage. I've seen it as a ton of different things in different textbooks over the time. Citric acid cycle. You know it as whose cycle? Maybe you don't know it. Anybody know another name for it?
Krebs cycle?
Oh yeah, Krebs cycle. Okay. I think some German dude. Mr. Krebs. Probably Dr. Krebs. Krebs cycle or the tricarboxylic acid cycle. For those that I know, we're waiting to get in on that one. Citric acid cycle and the electron transport chain. Glycolysis occurs within the cytoplasm. Glycolysis occurs within the cytoplasm of the cell, specifically the cytosol, but we're splitting hairs. Out in the cytoplasm of a cell, we don't require oxygen, but it is the first step of oxidative breakdown of glucose. Okay? Because I got to break down glucose into pyruvate. We'll see here in a second. Before I can get into the goodie bag of the mitochondria. The transition reaction, citric acid cycle, and the ETC, the electron transport chain, all three of those are basically occurring in the mitochondria at some location. Transition reaction and citric acid, if you want to write it down here on this slide, you can. Those two basically are occurring within the cytoplasm of the mitochondria, the matrix. As soon as I have pyruvate, that we'll see here in a second, it gets escorted into the mitochondria, and then that's when the reaction immediately starts to happen. The electron transport chain, it also occurs within the mitochondria. It just occurs within all of those folds. You see all those folds on the inside of the mitochondria right there. Those are called cristae. Okay. Called cristae. And that is where this thing is occurring, electron transport chain. All right. So these are the four kind of major parts, stages, okay, of oxidative breakdown of glucose. Glycolysis in the cytoplasm, intermediate stage or transition reaction, citric acid cycle, and the electron transport chain all occurring within the mitochondria, whether it be the matrix or the cristae, depending upon which one you're talking about. Again, for steps 2, 3, and 4 there, I need oxygen. If I don't have oxygen, can't go into the mitochondria. All right, so this whole first step is called glycolysis. Look at what the word is. Lysis means to break down, to destroy. What am I destroying? Glucose. Okay. So here with glycolysis, I am breaking down glucose. Again, it's out in the cytoplasm of the cell, and that does not require oxygen. As you can kind of see, there are a number of enzymes. They all have very specific names. I'm not going to ask you. Okay. I can't even remember. Is it truly chapter? I think it is maybe this chapter that actually does it in this book. This book and the phys book are very similar. So I apologize if I brain fart on chapters sometimes. But again, there in the book, I think it'll show you each individual step. I am not asking every individual step for glucose being converted into pyruvate. All right. I want you to know where these things are occurring. Cytosol here, cytoplasm, mitochondria for the other guys. I want you to know who enters. Who enters the first stage of glycolysis? Glucose enters. It's in its name. What do I get out of these stages? Okay. So for glycolysis occurs in the cytoplasm of the cell. What enters? Glucose. What do I get out? For every one glucose molecule, I get 2 pyruvate molecules. It goes from a 6-carbon structure to a 3-carbon structure. Pyruvate goes on to the transition. Okay. This will go on to the next step, the transition reaction. Okay, the intermediate stage, whatever the hell you want to call it. I also get a whopping waboom, 2 ATP. That's net. There was a gross production of 4, but you had to invest some money, 2 ATP, before you made any money. So 4 minus 2 is 2. So that's why I say net production. So we have a net production of 2 ATP. Fantastic. A whole whopping 2 ATP. And then you get these NADHs. I get 2 molecules of 2 NADH. These guys are going to the, and I'm going to shorthand it here, the electron transport chain. Those dudes right there are going to help us yield a huge chunk of energy. Okay. Because 2 ATP so far, that doesn't sound like a lot, and it ain't. Especially when you're making roughly around, depending upon the cell, anywhere between 32 to 36. Let's just call it 32 for now. Making 32 ATP. Okay. Now, if I have oxygen available, that pyruvate, it goes on to the mitochondria, undergoes the transition reaction, citric acid, electron, yada, 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 yada. NADH, yeah, goes and enters, all that fun stuff. Excuse me. If I don't have enough oxygen available, pyruvate cannot enter. Okay, so I can't undergo the massive yield. Oh, excuse me. That is inside the mitochondria. Okay, we convert pyruvate into lactate, lactic acid. This lactate or lactic acid has to be dumped out of the cell because it'll change the pH. Pump it out of the cell, into the blood, to the liver, jig-jog back into glucose, back into the blood, back into the tissue. Glucose can be absorbed into the cell, make 2 ATP, and if we have enough oxygen, awesome. If not, same thing happens again. Okay. Let's talk in terms of, do I have glucose? Okay. Pyruvate, and we'll see the chemical reaction on the next slide, has to be escorted into the mitochondria to do the rest of the work. Again, I made reference to this. Here are those words. Okay. The folds are the cristae. And this is where we're going to find the electron transport chain. The matrix is just the cytoplasm of the mitochondria. This is where we're going to have that transition reaction occur, as well as the citric acid cycle, Krebs cycle occurring. Okay. So we have a couple specific areas that, if someone's going to miss points and be nickeled and dimed points, generally that's what it is. Okay. It's the specificity of stuff. All right. If I can give you any advice that people usually say something that they didn't realize the first exam with me, is the specificity of things. Okay. When it comes to this thing, cellular respiration, cytosol or cytoplasm, and the mitochondria where these things are happening. But when it comes to mitochondria, there are different spots, different areas where these things are occurring. The matrix is one of them, the fluid of it. It's happening everywhere. It's not one specific, oh, it's happening right specifically here. Happening all over the fluid component of the mitochondria. In those folds, those cristae, that's where we find all of these electron carriers that make up the electron transport chain. That's where we find those. So that's specifically where the ETC occurs, cristae. In the matrix is the specific locale where the citric acid or the Krebs cycle is happening. Okay, so it's specificity that nickel and dime people on that first exam. There's the little reaction down there in that bottom right-hand corner there, as you can kind of see, pyruvate, a 3-carbon creature. Acetyl-CoA helps to bring pyruvate into the fold. We have acetyl-CoA, and we have an enzyme there called pyruvate dehydrogenase. Helps to bring acetyl-CoA and pyruvate together, or coenzyme A and pyruvate together. Am I going to ask you the enzyme? I don't know. Usually I don't. Okay. Usually I don't. But where is this thing occurring? Matrix, mitochondria. Who enters the transition reaction or the intermediate stage? Pyruvate, coenzyme A. That is what enters. What do I get out of it? Well, some good stuff. One of those things is acetyl-CoA. I have it under there. I have it underlined. Acetyl-CoA moves on to the next step, Krebs cycle. As you can kind of see from the picture there, I also yield a molecule of NADH. NADH goes to the electron transport chain, just like it did in the first step, in the first stage. Again, it's going to help to yield us a greater amount of energy. So I have acetyl-CoA. I have NADH. And what do I have left? I have waste. I can't do anything else with that sucker. Okay. So that is removed from the system. That is removed from the system. Another thing that may or may not be asked, sometimes it is, sometimes it isn't, is what's the yield in this intermediate stage? And you would say acetyl-CoA, NADH, and CO2. You'd be partially right, depending upon how the question is worded. Okay. What is formed? Yes. Okay. NADH, acetyl-CoA, CO2. But really, what's the yield of this intermediate stage? Because remember, one glucose molecule becomes 2 molecules of pyruvate. So what's going to be the real yield here? 2 NADHs, 2 molecules of acetyl-CoA, 2 CO2s. And I know it's semantics, and I know it seems like I'm being a DH. But sometimes that's the difference between getting a multiple choice question correct and not correct. All right. And I am just as guilty of doing it as anybody else is. here in this class or not in this class, last semester, future semesters, okay? I will read half of the question and be like, booyah, that's easy, bang. And then it's like incorrect. You're like, ah. And if I had just read the rest of the question, it would have said exactly what I needed to do to go obvious. It's obviously that's the right answer, okay? So what's the total yield of the intermediate stage or the transition reaction, guys? It's 2 molecules of acetyl-CoA, 2 CO2s, 2 NADHs. Who comes there? Well, pyruvate does, okay? But I have 2 molecules of pyruvate, all right? Depending upon how I ask it sometimes gives you a slightly different answer, right? So what's the point of me saying all that stuff? For you to read the entire question, okay? Because I know what's going to happen on this first exam. You're going to know it. You're not going to read it all. You're going to get it wrong, and you're going to hate me. And I don't want you to hate me, at least just yet. Okay, I'll at least wait a month. All right. Where does Mr. Acetyl-CoA go to next? It goes to the next step, next stage. That stage is Mr. Krebs and his little cycle. And there it is simplistically in all of its glory. Okay. Again, I'm not going to ask you I'm not going to ask you to go through every individual step here. Tell me which enzymes, what's the names. You don't need to worry about citrate, isocitrate, fumarate, alpha ketoglutarate. You don't need to worry about all that nonsense. Okay. I want you to know, again, where is this happening? The matrix, right? I was looking at the next slide. The matrix of the mitochondria. Who enters? Acetyl-CoA. It is a cyclical cycle because the end product is the first reactant. And that ultimately gets us the final product every single time. Okay. What enters? Acetyl-CoA enters. What do we get out of it? Well, I mean, you can, okay, yeah, I can see the picture there. There's some ATP, there's 1, 2, 3 NADHs, there's an FADH2, but remember, for every 1 molecule of glucose, 2 pyruvates. So everything is doubled up, right? So we get 2 ATP. We get 6 NADHs. We get 2 FADH2s. Two ATP and we haven't made a whole hell of a lot of ATP yet, and we haven't. Okay. The last step is the electron transport chain. And since we haven't made a whole buttload of ATP yet, you can bet your britches that this is where it is, and this is where it is. There are a number of, as you can see, steps within the cycle. Just for grins and giggles here, the B vitamins that we have in 5-hour energy help us to create some of these intermediates that are here within this pathway. Help us to make some components of the enzymes that are within this pathway. And they help to speed up Krebs cycle so we can make energy a heck of a lot faster. We can make all these NADHs and FADH2s. We could send them to the electron transport chain and we could make a whole heck of a lot of energy. Okay. We have amino acids that can jump in here as well. Okay. Hell, even triglycerides can come in and some of their parts and pieces can come in and give us intermediates in this pathway. So in the grand scheme of things, from a biochemistry standpoint, man, a ton of stuff happens here. Okay. And for this thing to keep turning, you need just a tiny bit of carbs. Okay. You need just a little bit of carbohydrate consumption. If we don't have enough carbohydrate consumption, I can't make oxaloacetate. I can't help to regenerate oxaloacetate, that end product. And citric acid cycle won't turn if I don't have enough oxaloacetate. So if I have too much fat entering in here, it's not going to be able to regenerate that oxaloacetate as effectively. And so your ability to make energy is going to stop and stagnate. So all of these fatty acid chains that I've eaten up, your body's going to turn them into fat. And then that's going to go around your midsection. And then that's when you're going to start disliking yourself. Okay? Because you can't fit into your stretchy pants anymore. Okay? So when it comes to citric acid cycle from a biochemistry standpoint, a ton of stuff happens here. And we could spend probably an entire lecture just on this little step right here, but we have other things to discuss. Okay, so where does this happen? Mitochondrial matrix. Who enters? Acetyl-CoA. What do you get out of it? 2 ATP, 6 NADHs, 2 FADH2s. The cell can utilize those ATPs, NADH and FADH2 on to the next. Where's the next? The electron transport chain. And whoops, there it is. There it is. There's that inner folds here of the mitochondria. And this is where you can see these little bitty electron pumps here, electron carriers. If I go over here, do I have, yeah, just a more simplistic view of it right here. Okay. Here's the glycolysis occurring outside the cytoplasm. This is all inside the mitochondria, obviously not to scale. As the pyruvate enters, it undergoes the intermediate stage. Again, the matrix. Acetyl-CoA. Sorry, guys. Didn't mean to do that into the mic. Acetyl-CoA, get out of the intermediate stage. Where does it go? Citric acid. Go in here. NADH, FADH2s, go to the electron transport chain, and NADH comes here and drops off those electrons. What does it drop off with the electrons? Protons. Okay. Well, electrons. Electrons get passed from one carrier to the next. As it does that, I'm pumping hydrogens from one side of the barrier to the other side of the barrier. And what does this create? Helps us to create a concentration gradient. This concentration gradient is made by taking the energy inside these electrons and using it to pump hydrogen ions against their concentration gradient to a side with more of them. That takes effort, that takes energy. And we can do that. We can pass these things around. There's like 3 sets of carriers, 4 sets of carriers really, but we can take them from one carrier to the next. Okay? And every time we take that electron and pass it from one carrier to the next, as it does that, we pump hydrogen ions across because we're sucking this little guy dry. It creates a huge pool of hydrogen ions on this side of this membrane. There it is right there. Now I've created this huge concentration gradient. Now I can undergo phosphorylation and use that oxidative phosphorylation to pump that hydrogen ion back across the barrier, going down its concentration gradient from an area of high to an area of low. And in the process, adding a P to a double P to make a 3P molecule, adenosine triphosphate. What happens after I've sucked all the energy out of that little bitty electron? It's just sitting there alone, feeling used. Who comes by? Oxygen comes by. We're inside the mitochondria, for God's sakes, right? It's the powerhouse of the cell. Oxygen's inside that sucker. Those hydrogen ions are sitting there. We take that little bitty electron and we can put it to good use. And we connect those two individuals together and we make water out of it. There's your water byproduct. So what were the end results of glycolysis, of cellular respiration? Way back a handful of slides, a few slides ago, C6H12O6 got us what? CO2 and H2O. CO2 is all this waste that I've been producing, yielding nothing at that point, so I can get rid of it. Water, waste, get rid of it. And in the process, I've made, as you can kind of see there, a number of ATPs. 3 ATPs for every molecule of NADH, 2 molecules of ATP for every molecule of FADH2. FADH2, 2 of ATP, really easy to remember. So I've made a good amount of ATP just in this ETC alone. And that's where the vast yield of energy comes from, is that specific locale. All of that I said is kind of listed down on this slide. Yeah. Electrons are delivered by the NADH and FADH2, pass from one carrier to the next, pumping hydrogen ions across as I do, and you can kind of see all that nonsense right here. Here's NADH coming in, drops it off to this electron acceptor, and it passes it along. And the process sucks that electron dry. Created a concentration gradient. Okay. Where I can then use this huge pool of hydrogen ions to turn the turbine. That is the ATP synthase complex right here. And as you can see, I can move that hydrogen ion back across the barrier there. And in the process, make ATP, oxidative phosphorylation. Where they occur. What goes in? What comes out? Those are fantastic questions that always get asked on the exam. Sometimes they get asked as multiple choice questions. Sometimes they get asked as short answer questions. Okay. But on average, around 34 ATP. It's actually like some cells produce 32.3 units of ATP. Some cells produce 36 units of ATP. There's a whole bunch of stupid math involved with it and more current research. So it's never a whole unit of it, but just men in black yourself and forget that I ever said that. Okay. Just go with these numbers for this book. Every book's different. I apologize. But oxidative phosphorylation happens in the mitochondria. And that big yield of it is going to be there within the electron transport chain. Okay. What is oxidative phosphorylation? NADH made in glycolysis gets pumped into the mitochondria or gets transported into the mitochondria. NADH that we made in the intermediate step, transported to the mitochondria. NADH that we made in the citric acid cycle in the mitochondria gets transported to the cristae of the mitochondria. And same thing here. So that's why you see all of those listed under oxidative phosphorylation. Because the yielding of ATP from those molecules happens only in the mitochondria. It doesn't happen outside of the cell. Or I'm sorry, outside of the mitochondria in the cell. Does not happen. Does not happen. Oh, man. I will not ask you a short answer question based on anaerobic respiration. I believe that's what these last couple slides are. I will not ask a short answer question. If you see it on the exam, it would only be a multiple choice question. As this pyruvate accumulates, I'm just going to go here to the picture and kind of talk a little bit here. As the pyruvate accumulates within the cell doesn't like it. So it will start to add things to it, adds hydrogen ions to it because NADH can't go anywhere, can't get in the mitochondria. So those hydrogen ions will disassociate from the NAD, and we will add those hydrogens that are disassociated to the pyruvate molecule. That is what gets us lactate or lactic acid, same basic thing. This lactic acid, this will be pumped to the liver. And made into glucose. Okay. This is called the Cori cycle. For those that know, I'll just put it in parentheses here. C-O-R-I, Cori cycle. Okay. So that's the Cori cycle. Once the liver gets the goods, that lactic acid or lactate, however you want to visualize it, we convert it back to glucose. Again, delivered back into the blood and sent it back to the tissues. Okay. Instead of 34 ATP, again, we're only getting 2 ATP. So we go from being around 33% efficient to about 2% efficient. Okay. So again, not very good. All right. So if you were asked to maintain that level of activity, your body would eventually you would just shut down. You wouldn't be able to keep going. Okay. Lactic acid, that sounds evil and bad. And lactic acid isn't evil and bad. Okay. Lactic acid, that's how I call it. Lactic acid is actually something that's really good for your muscles. To get into that anaerobic state, the more you do it, they've discovered over the last handful of years that athletes that get into an anaerobic state more often when they're training, their muscles become more effective at pumping out lactic acid, making it back into glucose and shipping it back. It becomes more effective at it. They also become more effective at utilizing oxygen that's there and preventing lactic acid production. So your muscles become more effective at utilizing the oxygen that you're there, keeping you in an aerobic state and not get in an anaerobic state as quickly. So your muscles become more efficient at utilizing oxygen. Okay? And it's good for development. Okay. Getting a little bit of stress in that regard of the production of something that's detrimental to normal functioning. That's how cells adapt. That's how tissues adapt and become more effective at doing stuff. So getting into that anaerobic state is not necessarily a bad thing at all, even though we always discuss it as, oh my gosh, lactic acid is so evil. That's not the case. Okay. If you're an individual who, is maybe, training for the first time, is overweight, obese, something like that, getting into an anaerobic state is one, not going to take very long. But B, you're going to be muscles are going to be really affected by it compared to someone who trains regularly. They're not going to be really affected as much to where somebody who is a very upper level athlete, they wouldn't feel probably effects at all. Okay. So it just depends on, how conditioned, how trained the muscles are, your cardiovascular fitness, all that fun stuff. I made reference to it earlier. Again, I'm not going to get in. I'm not going to get into that. If you're curious about this, come to my nutrition class. All right. The on-ground nutrition class, we talk about all of these macromolecules, where they enter into the metabolic pathways, cellular respiration, all that fun stuff. So, but fatty acids can enter into it. I made reference to it with the Krebs cycle, with beta oxidation, the use of fatty acids and whatnot. Fatty acids are huge molecules, huge chains of a triglyceride molecule. Okay. And we cut 2 carbohydrates off at a time. Well, if this thing is, 200 carbohydrates or 200 carbons long, sorry, that's a lot of 2s that I can cut off. And each 2 that I cut off yields acetyl-CoA. That's a hell of a lot of acetyl-CoA molecules, all showing up at the exact same time at the door to get in. That saturates the system. It bogs the system down. It'll start churning through all of those little molecules. And eventually, if you don't get enough carbs, that system can shut down. When you get in ketosis, you have so much acetyl-CoA there that the cell's like, oh crap, I can't. Dude, you dudes ain't supposed to be here. So it'll convert those molecules into 3 different ketone bodies. Okay? For somebody who's diabetic, who has an sugar imbalance, they can potentially not have enough, especially if they're type 1 diabetic, cannot have any carbohydrates, any sugar getting into that cell. So that really slows down the Krebs cycle. How do you know that that person is in ketosis or ketoacidosis for that individual? That fruity smell, right? Those acetones that they're exhaling. Okay. There's a couple other ones as well. One is exhaled. A couple of them are exhaled. One of them, it's very detrimental to the kidneys and whatnot. So it converts it right away into one of the other ones. So yeah. So when you're type 1 diabetic, it's easy to get that. You constantly got to monitor your sugars, that sugar balance within the blood, to really make sure that, because if you don't, the next source that the body's going to use is going to be fatty acids. And again, if I don't have enough carbohydrates to keep churning Mr. Krebs cycle, that could be a whole world of S word for that individual. Okay. Systems can start shutting down. Amino acids can be used in the same way to make energy. This is not optimal. You only utilize amino acids as your primary source for fuel as a last resort. Amino acids, proteins, they make things, structures. When you're utilizing, when you're breaking down structures, you'll get amino acid components to enter into the metabolic pathway at multiple different locations. To break down those amino acids, I got to clip off the nitrogen. The body doesn't have a way to store nitrogen. There's no nitrogen stores within the body. It's urea. It's gotten rid of. Okay. So we convert that nitrogen group we incorporate it into urea, and we just urinate it out. It's excreted through the kidneys for the most part. All right. But amino acids is a last resort. Okay. So then the question gets asked, well, in bodybuilding, I take my protein powder. Yes, if you take your protein powder after you're lifting, yes, you're utilizing it not for energy for the cells, but to help cellular growth and repair. Yes, you're flooding the system with leucine, isoleucine. I get it. All right. Yes, you're going to use it a little bit for energy, but ideally it's going in to repair the micro damage that you've done to yourself during the training session. What also do you do after you train really hard like that? you eat your carbohydrates. The best time to consume your carbohydrates, guys, is right after you work out. If you eat your carbohydrates more earlier in the day, your body's going to be still optimally burning, okay? Especially if you train. And I know I'm getting way off kilter, but this is fun stuff. This is how the body works for me. If you train certain ways like HIIT and Tabata, you keep your metabolism revved up and running for several hours longer throughout the day, 7, 8, 9 hours throughout the day. Okay, so consuming those carbohydrates in the earlier part of the day after you've trained like that, your body's going to utilize them, suck them right up into those tissues or whatnot, and utilize them instead of turn them into the F word. And no one likes the F word. Okay. And that's the human body. See, you're talking about one thing and bam, left turn, and then all of a sudden you're in nutrition class. It's a great thing about the human body.